Hello, my name is Abhishek Chavan. Today I am going to tell you about the clinical trials. The clinical trials is a systemic investigation in which the human subjects in which we use human subject for checking the safety and efficacy of the new drugs. The steps involved in clinical trials are: first is the preclinical trials, which is known as the animal study. In preclinical trials, this study is done on the animals. The preclinical trials are of two types: pharmacodynamic and pharmacokinetics. Pharmacodynamic studies, in which in vivo and in vitro studies are done, and pharmacokinetic means absorption, distribution, metabolism, and elimination of the drug we check. Then after preclinical trials, we used to fill the IND application form, that is known as the investigational new drug form, for the further phase clinical phase for the further clinical trial phases. Then phase 0. Phase 0 is nothing but the microdosing phase, in which study of new drugs in microdoses to derive the pharmacokinetic information in the human subjects. The microdose is less than 1 upon 100 of the dose of test substance calculated to produce pharmacological effect. Maximum dose is less than or equal to 100 micrograms. Main objective to obtain preliminary pharmacodynamic data. Then phase 1. In this in this phase 1, 20 to 25 healthy volunteers are involved. This phase is duration about 6 to 12 months. In this, patient of anti-cancer drugs or AIDS therapy are involved. The aim of this phase trial is to determine the maximum tolerated dose of the new treatment. Then phase 2. In this phase, 20 to 300 volunteers are involved, and the duration of this phase is 6 months to several years. And this phase is done to confirm the effectiveness, monitor the side effects, and further evaluate the safety. Phase 2 has the two types: phase 2A and phase 2B. Phase 2A is designed to access the dose requirement, and phase 2B is designed to study efficacy. Then another phase is phase 3. This is known as the therapeutic complementary trial phase. The target population in this is 100 to 3000. Duration of this phase is up to 5 years, and is used to establish efficacy of drug against the existing therapy in larger number of population. The phase 3 have subtypes. The phase 3A, in this phase, is aimed to get the sufficient and significant data. Then phase 3B allows patient to continue the treatment level expansion. Then after this phase, there are the after this end of phase, there are new drug application. The new drug application refers to the new drug application, the former proposal for the FDA to approve the new drug in the market. Then phase 4. The phase 4 is nothing but the post marketing surveillance. It has no fixed duration. It helps to detect the ADRs, drug interactions, and also explores the new users of the drugs. And objectives of the phase 4 is to confirm the efficacy and safety of the drug in larger population during practice, detect the unknown adverse drug reaction, evaluation of overship. And this is the clinical trials. Thank you.